Hello everyone, good evening. Today, let's begin today's class. So, in the previous lectures, we studied about different kinds of enzyme assays and enzyme catalyzed reactions in the previous class. And in this class, we'll study about inhibitors because inhibitors play a huge role in enzymology. Um, so today we'll learn about inhibitors and how to design different inhibitors and different approaches to design inhibitors. So let's begin. So what are inhibitors? Inhibitors are molecules which reduce the activity of enzymes. So inhibitors are molecules. They could be any organic, inorganic molecules. That molecules could be either organic or inorganic. And these molecules, they reduce the enzyme activity. Now, inhibitors could bind to the enzyme either reversibly or irreversibly. Okay, so the inhibitors binding could be either reversible or irreversible binding. So inhibitors which bind irreversibly to the enzyme, they actually modify the enzyme completely. There is a change in the conformation, and those inhibitors are known as suicide inhibitors. These inhibitors are widely used in enzymology, suicide inhibitors. They are basically used to study different enzyme function, how the enzyme actually catalyzes activity, what is the mechanism of enzyme catalysis. So these inhibitors are used for that enzyme studies, enzyme catalysis studies. And then we have reversible inhibitors. These bind reversibly to the enzyme and they do not completely modify the enzyme. So this is very important, that inhibitors should not completely disrupt or denature the enzymes, disrupt or denature the enzyme structure, that is our protein structure. Proteinaceous 3D conformation should not be denatured. And one of the important properties of the enzymes of the inhibitors is that they should be highly specific. They should be specific um, towards specific. They should be specific and selective towards the enzymes. Okay, so there are different different types of reagents that denature or that disrupt the activity of enzymes. For example, we have urea. So we have urea denaturation, and we have SDS, which is commonly used to denature enzymes. And this denature is pretty generic. That is, that urea and SDS will pretty much denature all the protein. Okay, all different types of enzymes. So they are not specific, or they are not selective towards one class of enzymes or towards one specific enzyme. Uh, so we cannot call urea and SDS as an inhibitor, even though they will disrupt the enzyme activity, but they won't be called as inhibitors. Okay, so we have enzyme that will bind to inhibitor. The binding could be either reversible. So this is the reversible binding. It will form an enzyme inhibitor complex as shown here. and it will reduce the activity, as in it will occupy the enzyme. And how does it occupy the enzyme? So enzyme has so if this is our enzyme, 3D structure of enzyme. So enzyme has a active site, okay, or this active site is nothing but substrate binding pocket. And this substrate binding pocket is the crucial part of enzyme, because due to this active site we have catalysis happening. And that's why the active site is very crucial. So in order to design the inhibitors, different inhibitors for specific to the enzyme, we need to have two information in we need to have enzyme and binding site information. We need to have enzyme structure information and its binding site information. And we should also know the inhibitor structure. Knowing the structure of inhibitor is also important in order to design different inhibitors. So there are different models which explain enzyme inhibitor interaction, and one of the model which is very well known is the lock and key model. So this model has the name lock and key because it uses the same it has its the analogy is same here, that the key has the same 3D conformation which is exactly complementary to the lock. Similarly, enzyme will have its substrate binding pocket or the active site will have a complementary interaction, will have a complementary structure, that is as shown here, a inverted triangle, that will be complementary to the substrate, okay, and the substrate will go and fit very snugly into this site as shown here, okay. And just like a lock and key, so the key has a shape, a kind of a unique shape here, and this key is specific for this lock because the lock has a complementary shape to this key. So so we have a snug fitting lock and key complex. Similar to that, we have an enzyme and we have a substrate which have a geometric complementarity towards each other, and they fit into and form enzyme substrate complex. But one of the main drawback in this lock and key model is that the is because of the rigidity and it's not flexible, okay. So because the enzyme structure is rigid and it is fitting the substrate very snugly, that means it is not flexible enough, and that's why many of the enzyme substrate do not follow this lock and key model, because the enzyme should be complementary to the substrate should be complementary to the transition state of the enzyme, that is it should be perfectly complementary to its transition state and not on the ground state. So because of this rigidity, there are weak binding interactions which doesn't allow the catalysis. So this is one of the drawback of lock and key, that the overall structure is quite rigid and not flexible, and there's not many enzyme substrate or enzyme inhibitor interaction follow this lock and key model. So we have another model. This is Koshland's model, also known as induced fit model. This model was explained by Koshland. And in the induced fit model, as you can see, the enzyme doesn't actually it is not actually it's not actually complementary it's not actually complementing the substrate, but it has a different shape here as opposed to the substrate. So whenever there is a complex formation, it is flexible enough. Okay, it's pretty much flexible enough to adjust the enzyme accordingly. Okay, so the enzyme is flexible enough to adjust the substrate here, and then this adjusting or this adjusting is happening in the transition state. Okay, and which increases the binding energy. Okay, and which further facilitates catalysis. Okay, is this is generally this is the basis of enzyme substrate interaction, which we studied in the previous classes. So in induced fit model, we have enzyme getting adjusted to the uh, substrate shape. Okay, and then it is converting the substrate into product. So this pink block is getting converted into blue as shown here, and the products are released. And then the enzyme, after the product is released, the enzyme is further it further goes back to its original shape. So this is the induced fit model, that basically enzyme is inducing or adjusting into the shape of the substrate. So this model explains lot of enzyme substrate and enzyme inhibitor interactions very nicely. So many of the researchers or enzymologists follow this induced fit Koshland model. Now, in order to design inhibitors, like I previously mentioned, we need to have the information of the active site, right? So active site is the site around 20 to 30 amino acids, or it could be as few as 3 to 4 amino acids long. Okay, for some protein it is around 20 to 30, or for and some enzymes it could be of 3 to 4 amino acids. And this active sites have different functional groups. It has functional groups, and thus it has different functional groups interact with the substrate, and then this interaction converts the substrate into product formation. So we have a product formation after interaction. So that's why having the information about the active site is very important. So there are different approaches to identify active sites. So we have biochemical approach. We can use biochemical approach to identify the active site. Then we can use bioinformatic approach. And finally, we can use different structural approaches. So we'll study about this in detail. Okay, so these are the three approaches used for determination of active site. So so let's study about yeah, so the first method was biochemical approach. So as the name suggests, it uses a different biochemical approach uses different experimental methods, like cloning, site-directed mutagenesis. So biochemical approach is further divided into two types. So we have amino acid modification. So we can either modify amino acids. Amino acid modification chemically. Okay, so we are using different chemicals to modify amino acids, and here also we are modifying amino acid by doing site directed mutagenesis. Site directed, or we could either do a random mutagenesis, both. So we have amino biochemical approach mainly focuses on amino acid modification. Okay, and this modification could be either by due to different chemicals, or it could be by mutation. And the mutation also is of two types. We could either use site directed mutagenesis, or we could use random mutagenesis, that is, we can randomly mutate. So in the chemical modification by chemicals, so we have different chemicals. So let's take an example here. So we have hexokinase enzyme. So what does this hexokinase do? This hexokinase converts glucose plus ATP. Okay, it will add a phosphate to the glucose. It will convert glucose to glucose 6 phosphate plus ADP. Okay, so basically it's a kinase, that is it will transfer the phosphate group from ATP to glucose. Okay, so this hexokinase has in its active site, it has a cysteine. Okay, it has a cysteine amino acid in the active site. So this cysteine amino acid is modified by a chemical known as iodoacetate. Okay, so this iodoacetate modifies the cysteine. It modifies the cysteine, and now the cysteine is no longer functional, so the enzyme is also not functional. Okay, and thus we can use this approach to identify the active site. So any active site which contains cysteine can be modified with iodoacetate by adding different concentration of iodoacetate, and then we can see the enzyme activity, like has it reduced the enzyme activity, because now the active site is not functional. Okay, because it's modified, the cysteine is modified, and the not functional, and it will reduce the enzyme activity. So this is one of the example of chemical modification of amino acids. Then we can do site directed mutagenesis. So in site directed mutagenesis, we can just modify cysteine. We can convert cysteine to let's say, in same example in hexokinase, we can convert cysteine to alanine. So cysteine has a disulfide bond. So with alanine, which is a hydrophobic amino acid, there won't be any disulfide bond formation in the active site, or there won't be any interaction, and thus the enzyme will not be functional again. So enzyme will not be functional. Similarly, we can do random mutagenesis, and in the random mutagenesis as well, there will be a random mutation. So let's say there is a mutation not in cysteine, but some other molecule which is interacting, and with the random mutagenesis we can find out whether the activity has reduced again. So random mutagenesis also follows the same principle. We have to check the activity again, whether the enzyme is functional or not, by randomly mutating all the amino acids. So with random mutation, one of the advantages that if we have no knowledge about the active site, like if we don't know whether cysteine or any other amino acid is present in the active site, we could just do random mutagenesis in that case, where we don't have any information about the enzyme or its active site. So we could just randomly mutate the enzyme and study the active site. So this is the biochemical approach to study the active site. Then let's move on to bioinformatic approach. So in bioinformatic approach, what do we do? In bioinformatic approach, what do? We have different sequences. We have sequences, and then we have homologous. We can find the homology, right? We can find the homology in the sequences from the databases. We have different databases from where we can get sequences, and then we can find homology. So uh, from sequences we can find homology of different motifs, domains, and also the whole 3D structure. Okay, so so the active site will have some 3D motifs, like there could be alpha beta alpha motif present in the active site. So we can find the homology between different motifs. Okay, similarly we can also find out different 3D structures. Okay, so example for this is again same. uh, so the example is same, that many kinase motifs, like in hexokinase we have the enzyme will have a kinase motif. Okay, so this kinase motif have some structural homology. It will have some structural homology. Okay, and this structural homology is exploited, so it will have some similarities with the other kinase motifs. Okay, other kinase motifs which are already present in the databases. Okay, these motifs are kinase motifs present in the databases, so we just have to match from those databases. Okay, so different kinase motifs. So we can say that if there is a homology of if there is a matching, so we can say okay, there is a kinase motif present in the active site. So this enzyme this enzyme is responsible for the kinase activity, and this is what actually is used in the structural approach as well. Structural approach. So there are thousands of databases which have different so like kinases, we have hydrolysis. So there are different motifs which are similar, which have a homology with kinases, hydrolysis, um, and other enzymes. Okay, other classes of enzymes, and we could actually match these 3D structures with our enzyme and find out whether our enzyme is, whether the active site has a kinase domain or a hydrolase domain. Okay, and if we know that our enzyme is hexokinase, we could actually search for docking with phosphate molecules. So the kinase domains will have a phosphate binding site onto them. So docking with a phosphate, doing a docking experiment can help us understand, okay, if the phosphate is docking onto the active site, then we can say okay, this enzyme active site involves some phosphate, it is a kinase or it is a phosphorylase, so there is some binding of the phosphate. So these all approaches are under the structural approaches in order to find the active site. So so now we move on to inhibitors design. So we have completed the enzyme, structure of enzyme. So structure of enzyme we all can know by different databases. Active site information can also be found out through biochemical approach, bioinformatic approach, and structural approaches by generating different libraries and designing different molecules which structurally match to the domains present. Now let's move on to inhibitor design. So there are four different approaches. We have a traditional approach, we have ligand based approach, we have receptor based approach, and then finally there is a receptor fitting. So we'll study about each of this approach individually. So there are in inhibitor designing also there are conventional approach and targeted approach. So in the conventional approach, it we didn't have much information of the enzyme and substrate structure. Okay, so uh, we are just randomly so we in the so now we have different databases and different library generated for different molecules for different inhibitors for the enzyme, but previously when there was not much cloning or there was not much databases updated, when we had no information about the inhibitor or the structure of enzyme, they used to use conventional approach. So in the conventional approach, we first do enzyme activity assay in order to determine what type of activity actually it is performing, whether it is doing a kinase activity or whether it is doing a hydrolases, whether it's a kinase enzyme or it's a hydrolase enzyme, or we can have so first we need to have idea about the enzyme. We should have the knowledge of the enzyme. So what all reactions the enzyme is catalyzing, and what type of reaction it is catalyzing, and how which what all substrate or substrate requirement is, and what all coenzymes cofactor involvement is required. So all that optimization has to be done in this conventional approach. Pretty straightforward, what we have learned from all the previous classes. Whether when we have no information about the enzyme or the structure, then we do screening of different active molecules. So the screening of active molecules could be by generation of library. Here we are using a structure based approaches which we studied previously, where we generate different libraries, and these libraries could be from natural sources, it could be synthetic or de novo library. So in order to screen different active molecules, first we need to generate different libraries, and these libraries could be of different molecules. It could be a peptide library, it could be a inorganic compound or some organic compound library. It could be also a DNA library, DNA or RNA library, mostly DNA library. Okay, so these are different ways of making different active molecules by using either natural sources or by synthetically making libraries, and then we screen these active molecules with the enzyme. Okay, so this the then we do a binding or validation assay. So we after screening we can so basically we screen out which are the best possible candidates from all the library which we have uh, screened. And one of the drawbacks is that we don't have information about the off target effects, whether actually this library is actually binding to that enzyme or it is doing something else. So after screening those peptides, there is very slight chances that we can improve that inhibitor. Okay, by and so in order to improve the inhibitors, we first need to have the information of the enzyme structure. So without that, there is a very slight chance that we can improve the inhibitor, and it is very time consuming, and because it's not that simple, it requires a lot of um, different experiments, different assays to be performed, and it is very time consuming, and also it is very costly because of the reagents and different techniques which we are using, and which is uh, not economical, and manpower is required. So to in order to perform different techniques, we need to have skilled person to perform different assays. So conventional approach has many drawback, and thus targeted approach is mostly now we use, because we have lot of information about all sorts of enzymes. We have databases present, and we have different libraries also, structured libraries, library inhibitor structures also present. So we don't have to do enzyme activity assay from scratch. Those will already be present in the literature. So we can just, for example, we know what hexokinase does. It adds the phosphate group on the glucose. So we don't have to find out that information. We already have that information present in the literature. And then we can also perform virtual screening. This is very important. We can perform n number of screening with of active molecules virtually, using different computational softwares and designs, and then we can study each interaction, how they are interacting by so after performing virtual screening, we'll get some hits, and then we can do some interaction study, like what those hits, what interactions there are occurring in those hits. And improvement is also possible, because we can improve the interaction, like we can improve the binding, we can decrease the binding energy, and we check all the thermodynamic properties, and we can improve the interaction. It's quite efficient because we can do n number of screening in this approach virtually, and it's economical. We don't use much reagents here unless we want to screen at biochemical level. So at biochemical level also, we once so basically once we shortlist few of the active molecules by virtual screening, those 5-10 molecules which are present, we can do a biochemical assay for those only, and not of thousands of libraries with active molecules present in the library. So it's quite economical, and manpower is required here also. So but yeah, it's quite feasible for at it's quite feasible to do. So this is an example of designing inhibitor by a traditional approach. So we have a parasite which is actually parasite which is the Plasmodium falciparum, which causes malaria, and this parasite infects the RBCs at different stages. So this is one of the stages, a ring stage. So we have a ring stage and we have a Shoizon stage. Okay, so this parasite infects the RBC at ring stage. Okay, and so we can inhibit this step from ring to Shoizon stage by using different phytochemicals. So here they have used different phytochemicals. So the performing this experiment is also quite simple. So we have first we synchronize all the Plasmodium falciparum to the ring stage. So first we infect the RBCs with the parasite, and then this parasite has different stages like ring, Shoizon, and other stages. So we synchronize them all at the ring stage. After synchronization, we incubate at different concentrations of different phytochemicals or different drug concentrations, and then we check for this transformation, whether the cells are going into Shoizon stage or whether there is inhibition. So if there is inhibition, all the infected RBCs will remain at the ring stage. So this is what we are seeing, that if they remain at the ring stage, mostly they will be uh, uh, mostly they will be pink and they will be live, so we can stain them using different dyes, and they will look pink, which uh, signifies life. And if they go into Shoijon stage, okay, they will be stained purple because they will mostly be dead. Okay, so dead parasite will stained purple. So this is how we can screen by staining the cells. So then we have different inhibitors ligand designing. So this is the ligand based approach. So in the ligand based approach, as you can see, we need to have this 3D structure of the biological target. Okay, so for example, we have receptor. So 3D structure like receptor based drug design. So we need to have a biological target, and with that knowledge we can design an inhibitor which can actually fit into that biological target. And then we can use pharmaco based drug design, where we can actually mimic different pharmacological drugs, and we can make similar looking inhibitors. The structurally they should be similar so that they could also mimic the same binding as these different pharmacological drugs are binding to the enzyme. Then we could use computer assisted drug designing and other approaches like molecular graphics, pattern recognition, and receptor fit. So we'll study one by one. So in pharmaco based approach, so we have a drug here. Okay, so this is the reference drug, and this is the dopamine. So this gray drug is actually binding to the dopamine. Okay, and it's inhibiting the dopamine. So we have this blue color drug which is actually matching to the reference gray drug, as you can see here. And this gray will now this blue will actually go, and it will similar fashion it will go and bind to dopamine. So basically we are mimicking the pharmacological structure of the drug. Okay, which actually drugs which are actually screened and which are actually going and binding to the dopamine, these are dopamine inhibitors. And with that reference molecule we design a similar looking drug, like the structurally and the 3D conformation is also similar, and we are expecting that this uh, mimicking inhibitor will also go and bind to the dopamine as same as the pharmacological drugs. Okay, so this is the main idea behind pharmacophore based approach. Then we have receptor based approach. So in the receptor based approach, what do we do? So we have, first we need to have a active site identification. So once we identify the active site, so as you can see this is the active site here, which is so these are the domains of the active site: 1 2 3 4 5 6 7 8 9 10. So these are the domains which actually surround the active site. Okay, then after that we grow ligand fragments, and by growing, this is happening virtually, receptor based approach, that we are growing ligand fragments which are actually complementary to this active site regions. Okay, this 1 2 3 region. So this square box, in this square box will grow our ligand which will be actually complementary to the this domains present in the active site. So if it is fitting, then we can change the fragment, but if it is not fitting, then again we can change the fragment and we can again start. But if it is fitting completely, okay, then it could be a potential drug, it could be a potential inhibitors which will go and fit into the active site. So for this approach also we need to have a active site information. As you can see here, this active site, so we have this red molecules, we can grow the molecules. By growing, We can add different functional groups that will interact with this active site, and we can design a inhibitor in such a way. Yeah, so in similarly in the ligand based approach, we have this is another example. We have a metalloprotein here. So zinc binds here to different amino acids. So these are the amino acids present in the active site. So so if we remove this zinc, and we are now growing a peptide here, okay, we are growing a ligand which is a peptide, and we can grow this peptide in such a way that it is interacting, that is, it is forming some hydrogen bonds with the active site, with histidine, with two histidine and glutamic residues present here. Okay, it is forming some hydrogen bond, it is interacting with the zinc complex. Okay, so we can design this ligand in such a way that it is snugly interacting with the surrounding molecules. And now this inhibitor, if it is interacting, it will be it will go and fit into it, and it will not allow the substrate to go and bind. So this is the main idea behind this approach. Yeah, so after once we grow a peptide, then we can further modify, we can do different modify that peptide into different functional groups. We can reduce the amino acids. So it's better to have a small peptide which can easily go into the enzyme. So we do different modification and then do fitting, and these are the different amino acids. So this H, histidine, glutamic acid, arginine, these amino acids are interacting with the peptide here, and now this peptide can be used as a inhibitor. Then we have computer based approach. So in the computer based approach, we do structure based drug designing. So we have a ligand database, just like our library in experimental conditions. We have different databases of different structural functional ligands, and then we have a target protein, and then we do a molecular docking. Okay, so what is molecular docking? We are basically fitting that ligand. Okay, based upon the molecular properties, the ligand will go and bind to the target protein, and that binding is known as docking. So ligand will get docked on the protein's active site, as you can see here. And then we study different pharmacokinetics and pharmacodynamic optimizations we do. Before this, we also study different molecular docking properties, like, so this is the molecular docking approach. We have target and we have ligand, and the ligand will go and it will snugly bind to a pocket in the target protein. Okay, so the molecular docking is used for the, so what is the rationale behind the design of drugs? So this molecular docking identifies the ligand's correct binding geometry. Okay, it will go and it will not bind anywhere else, it will selectively bind to the active site. So the ligand will be designed in such a way from the databases. It will be selected in such a way that it will have a correct geometry at the binding site, at the substrate binding site. Okay, and then we'll predict the binding affinity. So we'll have different scoring functions which will tell us how nicely the ligand is binding to the target. So molecular docking approach could be of protein ligand, it could be protein protein, it could be a protein nucleotide also. So we can have different ligands which could be either organic, inorganic molecules. We could have different metallocomplexes like cisplatin and other inhibitors. We can have different proteins or peptides. We can do molecular docking, and we can also do DNA RNA docking as well. It is also widely used. Okay, so this is the molecular docking approach, and so for this approach we need to have a target selection. So what is this target selection? It will be our protein with a binding site, with the substrate binding site or inhibitor binding site. We'll select the target first. We'll select that few amino acids surrounding the active site, like as shown previously, some glutamic histidine or cysteine which is present. So we'll select those amino acids, And we'll also do a ligand selection, and this ligand selection can be done from the databases, or we can make our own de novo library in the database. Okay, so it could be of both. You can either use already available databases, or you can make your own de novo ligand library. Then we do target preparation and ligand preparation, and then we do a docking, and then we evaluate docking results based and do different energy optimization. So energy optimization is done for to study the binding, how effectively a ligand is binding to the or inhibitor is binding to the protein, okay, or the target selection. So, docking is very widely used. It is used for drug targets, to identify different drug targets, whether the cancer drugs or different malaria drugs or COVID-19 drugs, how it is going and binding into the different pockets of enzyme, or if it is binding non-specifically to the enzyme. We can study all that using docking. Then we can also use docking to study different protein ligand interactions, um, whether it is binding non-specifically. Okay, so we can have better understanding by understanding the machinery of life. Basically, we have better understanding of enzymology, how enzyme works, antibody antigen interactions, and other different drug enzyme interactions or drug protein interactions. Okay, then for protein therapy, so many proteins or peptides are used as therapies for different diseases like Alzheimer's or other diseases. So we can use docking for that, and then we can engineer different protein enzymes which have better efficiency, or which has um, better binding towards some drugs. So we can engineer different protein enzymes. So the reliability of this docking method is not high, because it generally depends on the energy optimization and minimization. So it's all thermodynamically controlled, and it should be backed up with the experimental data. So this is very important. So docking is usually backed up with the experimental observations. Once you have docked and finalized your molecule, you can do experiments related to that, and you can have better understanding, and you can reliably prove that this docking method is reliable and good. Yeah, so if there are no experimental evidences, then it could be a false positive also, and it uh, because of different scoring strategy, it would give us false positive results. It will show very high binding, but when you actually do the experiments, you won't be able to get such high bindings. So it's very important to have um, uh, experimental validations of the docking results. And again, then applications are also very wide. So we can do a virtual screening, we can do a hit identification. So we have virtual screening of different libraries and peptide libraries, or different organic inorganic libraries or de novo libraries, which we can do a hit identification, and basically we can screen them. So we can screen ten thousands and lakhs of different ligands or peptides, and we can narrow it down to either 50 or 100, and those can be again narrowed down to 5 or 10 ligands, which can be further proven experimentally, because it's not possible to screen thousands of ligands do by experiments. It is very time consuming. It will require a lot of cost, and um, other it will require a lot of cost and manpower. So it's not that efficient. So it's docking is widely used in drug discovery, and it can also improve the drug binding. Okay, and we can have more so we have different generations of drug which are more potent, and So we can it is widely used in drug discovery and bioremediation. So protein ligand docking can also be used to predict different pollutants that can be degraded by the enzymes. So these are the applications for molecular docking. So let's move on to assignment. Let's move on to the assignment questions. Allosteric inhibition is a kind of. So what is allosteric inhibition? So we have enzyme. So this is an enzyme, and we have a active site. Okay, this is our active site. So allosteric inhibition is when the inhibitor will come and bind to a site which is not the active site. Okay, it is not binding to the active site, but it is binding somewhere else. So competitive inhibitions actually go and bind in the active site. So competitive inhibitors will go and bind to the active site. Okay, non-competitive inhibitors, suicide inhibitors will also go and bind to the active site, but they will bind irreversibly. That's the only difference. They will bind irreversibly. These all are reversible inhibitors. This is uncompetitive, also reversible. So uncompetitive is all will bind to competitive inhibitors will actually compete with the substrate and it will bind to the active site. Uncompetitive inhibitor, it will bind to active site and it will also bind to the both. It will bind to the active site and it will bind to some. Uncompetitive inhibitor will bind to active site and it will bind to some other site, which is the allosteric site. And the non-competitive inhibitor will go and bind to the other sites. The correct answer is B, non-competitive inhibitor. So allosteric site is site other than the active site. Inhibition of invertase by sucrose is dash type of inhibition. So invertase is an enzyme. It converts sucrose. Invertase, the substrate of invertase is sucrose. Okay, it converts sucrose into other sugars. Okay, other sugars. This inhibition is actually a substrate inhibition because sucrose itself inhibits invertase. Okay, so this inhibition is known as substrate inhibition. So if other sugars like inverted sugars, if it inhibit invertase, then it could be a product inhibition, but sucrose itself inhibits invertase, so it's a product inhibition. Match the inhibitors and of the protein enzyme with its target disease. So these are the different target diseases: human renin, anti-hypertension, collagenase, anti-cancer, purine nucleotide phosphorylase, antidepressant, thymidine synthase, anti-proliferation. So the correct combination is this A1B2C3. This is the correct combination. Autodoc, in Autodoc the term grid refers to, so the grid refers to the 3D space surrounding the target proteins where the docking is performed. So this is important. The grid refers to this 3D space of the target protein where the docking is performed, and this 3D space mostly will include the active site. It will mostly include the active site. It will be surrounding the active site. In drug design, targeting the active site of protein is important because it allows specific interaction with the substrate molecules. So It is very important to target active site because that's the site where the substrate will interact the most. So it would be better to have a specific interaction. It will target the active site. Correct answer is B. The term pharmacophore is in inhibitor drug design refers to um, the active site of enzyme, a class of reversible mechanism based inhibitors, essential features of drugs that contribute to its biological activity. So the correct answer is D. So in pharmacophore based approach, we are basically mimicking pharmacological drugs. Okay, we are basically mimicking those drugs for the activity. So if we have a drug like this with different functional group, we are actually mimicking the same drug. Okay, we can add few functional group in order to improve the potency of the inhibitor, but basically we are mimicking the same structure so that it will be complementary to the it will show same biological activity. Suicidal inhibition, also known as mechanism based inhibition, involves, so as I mentioned, it involves irreversible inhibition of the enzyme. Okay, it irreversibly binds and completely modifies the enzyme, and it will change the enzyme structure. The main information obtained from NMR spectra in structural biology is mass of molecule, three-dimensional structure, absorption of visible light, surface area of molecule. So basically NMR is used to find out the structure, like what all structure or what all functional group is present. So with NMR we can find out the complete structure of molecule. So how much spectra this carbon is emitting because of its bound to OH. So basically it will give us the 3D three-dimensional structure. Initial so this is the problem. Initial velocity for the enzyme catalyzed reaction is 4 micromole per minute, and the substrate concentration is 2 millimolar. If the enzyme concentration is 0.5 micromolar, the calculate Michaelis constant. So we have initial velocity to be 4 micromole per minute, and we have substrate concentration 2 millimolar, and then we have enzyme concentration to 0.5 micromolar. We can convert this into micromolar only, so this will become 2000 micromolar from milli to micro. And we have to calculate Km. So we can just directly use Michaelis-Menten formula, where V0 is equal to V max substrate concentration upon Km plus substrate. So here instead of V max we have total enzyme concentration they have given us, and we know that V max is equal to ET and Kcat. Okay, so in order to substitute V max we need to have ET and we need to have Kcat information, and this Kcat information is actually missing from here. So because of this we cannot calculate this, okay, because we don't have information of Kcat here. So hence we cannot calculate V max, and thus we cannot know Km. So the information is missing in this text. Question: in receptor based drug design, the term allosteric inhibition refers to inhibition of enzyme active site, inhibition of binding to site other than active site, inhibition of drug absorption, drug metabolism. So yeah, allostery means binding to the site other than active site always. So allosteric inhibition is inhibition of binding site to other than the active site. Simple. The process of designing inhibitors that mimic the structure of natural ligand for target receptors is reverse pharmacology, structure based drug design, ligand based drug design, de novo drug design. So basically we are mimicking the structure of the natural ligand, that is structure based drug design. Okay, correct answer is B. For enzyme catalyzed reaction, V0 initial velocity is 6 micromole per minute at substrate 0.5 millimolar and 2 micromole per minute at substrate 0.2. Calculate the slope and y-intercept of Lineweaver-Burke plot. So this is our Lineweaver-Burke plot here. It's a reciprocal of the Michaelis-Menten. And the slope which we get from that Lineweaver-Burke plot is, the slope which we get is Km by Vmax, and the intercept here, this is the intercept which we get is equal to 1 by Vmax. Okay, so we can calculate, we can plot this graph. So I have already calculated here. So whenever so if we have 500 substrate, this is how the graph will look like, and the slope, the intercept here 1 by V, so the intercept here 1 by V max is equal to 1 by 0.167. Okay, this is the intercept, this length, which is approximately equal to 6 micromole. Okay, similarly the slope will give us Km by Vmax, and the slope is 0.11. So yeah, so the correct answer is the slope will be Km by Vmax and the y-intercept will be 1 by Vmax. The scoring function in molecular docking refers to determining the similarity between the ligands, evaluating the fitness of ligand conformation and binding, calculating the molecular weight of ligand, estimating the melting temperature. So no, we don't estimate any melting temperature or molecular weight. It is used to evaluate the fitness, like how good the ligand is binding to the conformation. Okay, how good the binding is, how much. So the fitness score will basically tell you how good the ligand is binding out of all the libraries which we are analyzing. In uncompetitive inhibition, the presence of inhibitor affects Km and Vmax as follows. So in uncompetitive inhibition, the Km is basically decreased. Okay, the binding is reduced. The binding is increased, and the Vmax also increases. So in uncompetitive inhibition, the Km decreases and Vmax increases. Correct answer is B. The software used for molecular docking studies is Photoshop, AutoCAD, PyMol, Autodoc. So we use Autodoc. Okay, so that's all for today's class.